So Damian Marron of TxCell is our next presenter. Damian, I'm going to let you kick it off with the slides, and as I always do, I'll interrupt throughout.
Thanks a lot, Jason. So great to be here. We're a public company, so classic disclaimer statement about any forward-looking statements that I may make. TxCell, we're a clinical stage personalized T-cell immunotherapy company. Clinical stage, we have already Phase I/II results in severe refractory Crohn's disease, and we are about to enter the clinic in non-infectious uveitis. So our products are directed at severe chronic autoimmune diseases. We're based in France. We've been around for quite a long time, which means we've got a great knowledge of what we're working on. We have now about 63 employees, a very strong patent estate. As you can see, we have our own GMP-certified manufacturing facility over in France. We had net cash at the end of 2014 of what would be about 16 million euros at current exchange rates, and a recent market cap that's in the $75–80 million region. I just want to talk a little bit about how our approach works with autologous, antigen-specific, type I T regulatory cells, which we use, as I said, to treat severe autoimmune diseases. So we create these antigen-specific type I Treg cells through our proprietary manufacturing process. When they're injected intravenously, the cells will naturally migrate to sites of inflammation. It's a characteristic of the type I Treg cell. The type I Treg cell is a tolerogenic cell of our body made to help us cope with endogenous and exogenous antigens. At step 2, the cells have migrated into the tissue. If they then encounter the site-restricted antigen that we have manufactured them to react to activate with, which is located only at the lesions, the cells will activate and they will proliferate. And then you go to step 3, where you unleash the power of a type I Treg cell as an anti-inflammatory cell. And that is through multiple molecular and cellular targets, release of anti-inflammatory cytokines, cell-to-cell interactions down-regulating pro-inflammatory T cells, actually killing other pro-inflammatory cells such as monocyte dendritic cells, and down-regulating the metabolic environment, the inflammatory metabolic environment. Then very importantly, about 10 to 12 weeks after they're injected, the cells are cleared. These are terminally differentiated cells. They create no memory. They're cleared away, which means that we don't have any issues, safety issues of long-term cells being around. It also says that if we're going to treat chronic diseases, we are going to need to re-inject. And indeed, from our first trial, we know very well that the duration of the activity corresponds perfectly with the lifespan of the cells in the tissue. So when we put all those things together, we have seen already high efficacy. In our first trial in severe refractory Crohn's patients, refractory to steroids, to immunosuppressors, to biologics, at the end of that, we saw 75% response rate in the best dose and 40% remission rate, which are pretty remarkable numbers in those sort of patients. Tolerability, excellent. These are a tolerogenic cell type. We don't even get into the issue of cytokine storm, for that matter. And we expect them to have low resistance because of the multiple pathways being difficult to overcome altogether. So just to wrap up at this stage, we are making what we believe is scalable and cost-effective personalized T-cell immunotherapies for these autoimmune diseases. We have a product platform. We can apply it by using different antigens, different site-restricted antigens for different diseases. We can treat a whole number of diseases. I've not shown you our pipeline here, but it has 2 clinical projects and 3 research projects already. With our lead product, Ovasave in Crohn's disease, we've already shown very good data. We're now in Phase IIb, and that is really an underserved market. The patients, once they've failed on biologics, have nothing today except surgery, which is only palliative. We have a partnership on that project with Ferring and Ferring's advanced therapy development affiliate, Trizell, which validates our approach here and provides a lot of lifting power to also things like the process development and the setting up of manufacturing, a process which we can replicate for our other products later. Ourselves, we're focusing on the orphan indications. Non-infectious uveitis, as I said, is just about to come into the clinic, and in the pipeline we have other orphan and niche targets, products which we intend to take through to registration. And our business model, we believe, is unique in this area. I haven't really spoken at all about the manufacturing, but we have to manufacture only one time from one small peripheral blood sample to create over 5 years of treatment for a chronic disease on the basis of treating every 8 weeks the patients. That obviously leads to a very attractive cost of goods and strong margins, where we're looking at biologic-type margins, basically. So thanks, Jason.
Good. So can we wind a few slides back? Let's go back 2 slides. I want to talk a little bit, just make sure that I'm clear. Yeah, let's start one more forward. Let's go to that. Yes, please. I just want to make sure I understand the business model. So we're talking about manipulating the T cell population, and I think about T cells as T effector cells and T regulator cells. So when you start with a patient, first of all, we're talking about an autologous therapy, right?
Absolutely, yeah.
So
absolutely.
and the patient is doing a blood donation or an apheresis, is that right?
Yeah, let me flick forward. I've got a few slides back here. Here we go.
And while you're doing it right, so help me understand exactly what you're taking from the patient.
Yeah.
Here's the direction that I want to go. How do I know, let's say, a patient who has Crohn's disease, a disease that waxes and wanes? And you know, I happen to believe that multi-stem and allogeneic stem cell therapy probably works in Crohn's disease, even though the Pfizer ulcerative colitis trial failed. It'll be very interesting to see what happens with results from the Osiris, today Mesoblast prochymal trial that's running in Japan around Crohn's disease. So the question I have is, though, that, you know, this is a very tricky trial to run because the disease state changes. And what happens when I take something ex vivo, and how do you manipulate it, and then I put it back in the body? How can I be assured that it does what I think it's going to do and doesn't, say, change its characteristics in vivo versus ex vivo?
I mean, there's a whole bunch of really great questions there in what you've just gone through, Jason. First of all, what we do, so just to talk quickly through this slide, we start with a peripheral blood sample, classic separation of the leukocytes, as you might expect, and then we enter the proprietary part of our process, where we stimulate those cells with the antigen that we want. So we choose, as I said, antigens on the basis that they're going to be site-restricted to the lesions of the disease we want to treat. And then different antigen, different disease. So actually we use ovalbumin for Crohn's disease, food antigen, that will be found distributed in the tract after you've eaten. For non-infectious uveitis, it's collagen type 2, which is localized in the eye. Nothing to do with the disease, but it's just there, and it will then therefore be able to activate the cells. We get an enriched population, and from that enriched population, we have knowledge of the phenotypic characteristics and other characteristics of the antigen-specific type 1 Treg cells that we want to use as the treatment. That allows us to take them out from that pool of cells and have a pure population of antigen-specific type 1 Treg cells.
All right, so let's back up a little bit. So what you're doing is you're exposing the T cells, but when we talk about T cells, you're talking about activated T effector cells?
We are talking about any T cells that will react to that antigen. So you will have T effectors, you'll have Tregs, you'll have, you know, various subtypes of all of those.
And are you desensitizing them? Once they're activated, it seems to me that if I take T cells and I activate them to the antigen, you could tell where I'm going, that if I put them back into the body, how come I'm not exacerbating the disease condition?
Because, as I said, we have the technology, the knowledge to be able to separate out just the AG Treg cells, the type 1 Tregs. All the others are discarded in the production process. So we have a pure population at the end. It is also a terminally differentiated population of cells, and we have tested them to death to look for plasticity, the risk of them switching over to a Th17 type phenotype. That doesn't happen. The regulators have also been on our backs about that, of course, as you would expect, but we don't see it.
So what I hear you saying is that you're essentially changing you're taking out everything that reacts, and you're putting back in all the things that don't react. So you're maintaining T cell immunity, right? But you're not. But you've desensitized that population to that antigen. Is that correct? In the hopes that it
no,
then
we have
doesn't...
sensitized that population to that antigen, that Treg population to that antigen. We want them sensitized to it so that when they're injected and they go to the inflammatory site and they encounter the antigen there that they're sensitized to, that they will activate. We want them to be actually sensitized to it and to activate there at the site, but only at the site and only in the presence of the antigen.
But if they activate at the site, how come I'm not locally exacerbating the disease condition? So in the case of ulcerative colitis, right, I have an autoimmune problem at the site of the colon.
Well, I mean, if I talk more about Crohn's, you mentioned UC there, but I prefer to stay on Crohn's where we're really pushing forward the development. Crohn's, like so many of these autoimmune diseases, is actually characterized by a dysfunction of the Treg compartment. There's a lack of, or they're not fully functional. And your immune system, putting it into most basic, is out of balance. And what we're doing by putting back fully functional Treg cells, which then activate at the site of the lesions, is we're bringing that back into balance. And these are anti-inflammatory cells. That is their that is their activity. And so they will treat the inflammation at the site.
Right. So I think that's the key here, is you're taking an activated T cell population that's kind of swung too far into activation mode, and you're returning it back to center. But how do you know that that it doesn't switch and become a hyper-activated? And obviously, there's some chemistry.
There's the 2 points to that. As we spoke about, first of all, the cells themselves we know are not plastic. They won't switch to a pro-inflammatory cell in themselves. And because they're targeted only to be activated at the site of the lesions, they'll go elsewhere in the body, but they won't encounter the antigen, so they won't activate. We have a local activity. We don't have any systemic immunosuppression, and that, of course, is something we've looked for very clearly as well.
So now, Damian, share with me, put on your business hat, and share with me a couple of things. What are the cost of goods of this process, and how do you get a Crohn's disease or ulcerative colitis trial done? It seems like it's a very, very tough trial to do.
Yeah, sure. Business model first, and cost of manufacturing and cost of goods. At the moment, this is a relatively long process that is not suited to the market. We know that, but we also know, and we are working on it right now, how to take this down to a process that will fit the medical practice. And when we get to that point, as I mentioned just before, we will have a margin, a cost of goods and a margin that is similar to a biologic product. So if we were to price Ovasave for Crohn's disease at the same price as existing biologics, we would also have the same gross margin as those biologics. So from that point of view, things are very nice.
And close with me a little bit on trial design.
Yeah, exactly. Doing a trial, and actually I can flick through again just a couple of slides. That's our results from previously.
You have 1 minute.
I've just gone too fast. No, here we go. Sorry. There we go. This is the design of the current trial. The key for us here is that we're working with the severe refractory patients with active inflammation. So this is a very defined population. Not that easy to find. So you can see to recruit 144 patients is going to require us 32 centers, and the timelines of the trial is about 2 years long to get from the start to getting the results. But the real key is to use that very chosen moderate to severe population, which is the population that really has the medical need in Crohn's disease. If you're mild or you're further back, you're easy to treat. If I say a point about what has been often the killer for Crohn's disease, placebo response.
Yes.
If you look through the literature at these type of patients, the highest placebo response you will ever see in the literature is 20, very rarely 25%. So what we have done in our trial is we've actually sized the trial for a 35% placebo response to really take out that risk we feel that we are going to, find our legs cut from underneath us with an oversized placebo response.
All right, and I see two other things that you're doing that's a little bit different. You're doing multiple injections, which I like.
Yeah.
And what's the time point on the readout for the endpoint?
The primary endpoint is actually being read at 6 weeks. So 6 weeks after the first injection is the primary endpoint. And then we have secondary endpoints after the second injection, and so on.
So it's very, very exciting. Yet another T-cell therapy in the space. A very interesting disease proposition, too. Thank you for joining us today, Damian.
Thanks a lot, Jason.